In this talk, we're going to learn how to analyze experimental data. The scientific method is a method that's used to obtain information by experimentation. It is a very logical method with a stepwise scheme. So we normally begin by making an observation, which is based on any phenomenon that the scientist finds interesting. The scientist then researches the topic, and he will go to reliable sources of information, like scientific articles. Once the scientist knows about the topic and the phenomenon that he's interested in, the scientist will go ahead and ask a question, which is basically what the scientist wants to know about the phenomenon that we don't know. Once the question has been asked, a hypothesis is formulated, which is a possible answer to the question that has been posed. Experiments are designed and performed, and this is usually done to test the hypothesis. Once the experiment is performed, the results that are obtained are analyzed, conclusions are drawn, and the scientist will document all his findings. Based on the experimental results, if the hypothesis is not supported, then it is disproved, and the scientist will go ahead and formulate a new hypothesis and go through the whole method again. On the other hand, if the experimental findings support the hypothesis, then the scientist is now ready to ask another question and go through the scientific method once again. Let us now look at what is a hypothesis. Hypotheses are possible answers to questions, and these help explain the observations. It should be remembered that hypotheses can be right or it can be wrong. The word hypothesis is the singular version, while hypotheses is plural. It should be remembered that while writing a hypothesis, it is written as a statement and not a question. When experiments are performed, a hypothesis can be easily disproved, but it is very difficult to prove a hypothesis because there may be some other explanation that the scientist did not think about that is also able to explain the experimental findings. Hence, instead of proving a hypothesis, scientists will accumulate evidence that supports their hypothesis. There are certain requirements for a hypothesis, like it has to be written as a statement and not a question. When a hypothesis is written, it has to be written in such a way that it is testable. This occurs when the hypothesis is well defined and narrow enough to give meaningful data. For example, stating that some chemicals kill cancer cells is a hypothesis, but it is so broad that it is difficult to come up with meaningful experiments to test this hypothesis. On the other hand, vitamin D lowers survival of cancer cells is also a valid hypothesis, and it is testable too, and thus it is a much more meaningful hypothesis. The hypothesis that is proposed should be measurable and controllable. For example, you can't have a hypothesis on what someone thinks because it cannot be tested. However, you can measure the effect of vitamin D and its derivatives on cancer cells. When we look at the different types of hypotheses, there are two main types. We have the alternative hypothesis that is denoted as HA or H1 or H2 and so on and so forth. And this is the hypothesis that is used to test through your experiments. It will usually state that there will be an effect on whatever you're studying, and hence it helps with the experimental plan. Thus, as in a previous example where we said that vitamin D lowers the survival of cancer cells, it is stating that there is an effect, which is the lowering of survival. This helps us design the experiment where we're going to look at the survival of cancer cells in the presence of vitamin D. The second type of hypothesis is a null hypothesis, which is denoted as H0 or HO. This is the hypothesis that states that the factor that you are testing has no effect. and it is prominently used for statistical analyses. Thus, for our alternative hypothesis, where we stated that vitamin D lowers the survival of cancer cells, the corresponding null hypothesis would state that vitamin D has no effect on cancer cell survival. It should be noted, to have a null hypothesis, the experiment should have numerical data or quantitative data. Now that the alternative and null hypotheses have been proposed, the experiments can be designed. Thus, the experiments are designed to test the hypotheses, especially the alternative hypothesis. Once the experiments are designed, we can perform the experiments and obtain data. This data that is obtained has to be analyzed. The data that is obtained for the different conditions and the replicates for each condition has to be analyzed. Since you have replicates, the central tendency, or a value that summarizes all the replicates of a condition, has to be found. This is calculated by finding the mean, the median, or the mode. Now, which central tendency should be chosen depends on the type of data. For the kind of data that we normally obtain in the lab and in vitro studies, the mean and the median is a commonly chosen central tendency. In addition to the central tendency, we also need to find the dispersion, which is how close the replicates are to one another. There are many different types of measures of dispersion, including the range, the variance, the standard deviation, and the standard error of the mean, or the SEM. Once again, for the type of experiments that involve in vitro studies, a lot of times standard deviation and standard error of the mean are the chosen dispersion variables. So now that we have our data analyzed, we can graphically show our data, as shown in this figure, where we're looking at the cell survival in cells that were not treated with anything or the no treatment, and the cells that were treated with vitamin D. As we can see in the graph, it looks like the bar that represents vitamin D is lower in height or shorter than the no treatment sample. Now, how do we know that this difference that is observed between these two groups is genuine? And for that, we have to perform additional statistical tests. Statistical tests are available to determine if there are significant changes among the different groups of your experiment. It is basically a measure to try to find whether these differences are genuine or they're occurring due to some random chance events. These statistical tests normally test the null hypothesis, and the null hypothesis in our specific example was that vitamin D has no effect on cancer cell survival. Now, the statistical tests that are used will analyze the data and calculate a probability value called as the p-value. The probability value, or p-value, determines whether the difference between the groups is by random chance. So when we look at our data and we find the p-value, if the p-value, which is a number, is more than 0.05, then we conclude that there is no statistically significant difference between the two groups. And what that means is that we will accept the null hypothesis, which stated that there is no effect of vitamin D on the survival of cancer cells. On the other hand, if the p-value that is calculated is equal or less than the cutoff of 0.05, which is about 5%, then it shows that the differences among the groups are statistically significant. And in this scenario, we will reject the null hypothesis, and thus we will conclude that there is a change in the cell survival when the cells are treated with vitamin D. When we do have a statistically significant difference between two groups, we either show that as showing the actual p-value that is shown in the figure, or we replace it by putting an asterisk. Sometimes other symbols can also be used, but an asterisk is a very common symbol that is used to show that there is a statistically significant difference among different groups. There are different statistical tests that are available to the scientists in order to calculate the p-value. Now, which test should be chosen, it depends on different factors, including what is the scientist studying, is the data that has been collected showing a normal distribution, and how many groups are being compared. Let us look at normal distribution of data. To know whether the data shows normal distribution, you can take all the replicates of a given condition, and when they are plotted, then we should get a bell-shaped curve. If you don't get a bell-shaped curve, then the data does not show normal distribution. Now, instead of just eyeballing the curve and determining whether the data shows normal distribution, we can determine the distribution of data by doing some calculations. In order to find normal distribution of data, all experiments should be done multiple times and in replicates so that there are many readings for a given condition. For every data set, we can find the mean, median, and mode. And if they are equal, then the data shows normal distribution. However, there are going to be many instances where the mean or the median and the mode are not equal to one another. And these differences could be minor. So how do you determine then whether the data shows normal distribution? In such scenarios, there are additional tests that can be performed, including QQ plots, the Kolmogorov-Smirnov test, and the Shapiro-Wilk test. And all these tests can be used to determine if the data shows normal distribution. If the data shows normal distribution, then parametric tests are performed to calculate the p-value. There are different types of parametric tests, and if there are only two groups involved, then a t-test is commonly performed. However, if there are more than two groups involved, then the parametric test that is commonly used is an analysis of variance, or ANOVA. It should be noted that when a t-test is performed, the t-test directly provides the p-value. However, when an ANOVA is performed, the p-value that is obtained simply states whether there is a statistically significant difference among the groups, but it doesn't tell you between which groups do we observe statistical significance. Thus, when an ANOVA is performed, post-tests like the Bonferroni test or Tukey test have to be performed to know which two groups or which other types of combinations of groups show statistically significant differences. If the data does not show normal distribution, then non-parametric tests are used to calculate the p-value. These include the Mann-Whitney and the Kruskal-Wallis tests. Now that we have a good foundation on central tendencies, dispersion, and p-values, let us analyze certain scenarios. We're looking at the experiment where the alternative hypothesis stated that vitamin D lowers the survival of cancer cells, and the experiment was performed to test this alternative hypothesis. Once the data was obtained, statistical analyses were done, and these statistical analyses are going to be testing the null hypothesis, which states that vitamin D has no effect on the cancer cell survival. Let us assume that this is the data that we got, where the p-value is 0.0565. Since the p-value is more than 0.05, we will accept the null hypothesis, which stated that vitamin D has no effect on cancer cell survival. Thus, we reject the alternative hypothesis, and the alternative hypothesis is disproved in this scenario. Let us now look at another scenario where the p-value that was calculated between these two groups turned out to be 0.0365. Since the p-value is less than 0.05, we will reject the null hypothesis. Now, to determine whether the alternative hypothesis should be accepted or not, we have to look at the actual trend that our experiment shows. Since vitamin D treatment decreases cell survival, and that is what our alternative hypothesis stated, we will accept our alternative hypothesis, stating that vitamin D lowers the survival of cancer cells. Thus, in this scenario, the alternative hypothesis is supported, and we can observe a statistically significant decrease in cell survival when the cells are treated with vitamin D. In this scenario, the p-value is still 0.0365. So since it is less than 0.05, we will reject the null hypothesis. However, when we look at the trend that the data is showing, we're actually seeing an increase in cell survival when the cells were treated with vitamin D. Since vitamin D increases the cell survival, it contradicts the proposed alternative hypothesis where we stated that vitamin D actually lowers the survival of cancer cells. Thus, we will have to reject our alternative hypothesis because the experiment does not support the hypothesis that we had proposed. In this case, the null hypothesis has been rejected and the alternate hypothesis has also been disproved or rejected. So in such situations, the scientists will formulate another alternative hypothesis and then proceed with their experimentation. Thus, in this way, we use different statistical analysis tools in order to analyze our data.